This is Lab Report 8, Biochemical Test Part 2. This is group number 2, Diego Castillo and Rita Cantu Mahurin. In the introduction, we highlighted the purpose as well as the key concepts of this lab. So we identified and differentiated gram-negative bacteria based on their ability to decarboxylate amino acids using lysine and ornithine, which aided in bacterial classification and metabolic profiling. As far as the key concepts, decarboxylase enzymes remove the carboxyl group from amino acids, so positive reactions indicate amino acid utilization and alkaline byproducts. The materials we used as far as media were the lysine iron agar, also known as LIA, and then ornithine broth. The bacterial cultures were E. coli, C. ferendi, E. clocae, Morganelli morgani, P. mirabilis, P. aeruginosus, S. typherium, and then our unknown or gram-negative isolate. And so as far as the equipment, we utilized the inoculating loop and needle, and then the incubator, and then our labeling materials, as well as our gloves. So as far as the procedure, it varied as far as the different techniques used per test. So the lysine decarboxylase test, we did stab the butt and streak the slant of the LIA tube with the test organism. And then we incubated at 35 to 37 degrees Celsius for 24 hours. In the case of our specific lab, it was a bit longer until the next lab time. And then we observed the color changes in the butt and slant and we recorded our results. And then as far as the ornithine decarboxylase test, we inoculated this specific broth with the designated organism, incubated it as well for the same amount of time until the next class time, and then we recorded the color changes and we also interpreted the results. And then as far as notes, stabbing ensures that there's anaerobic conditions, and then streaking tests are showing the aerobic growth. The purple color was a positive result, so it's an alkaline reaction, and then the yellow color was a negative result, which indicated an acidic reaction.
These are the results for lysine decarboxylase. What's the purpose of lysine decarboxylase in gram-negative bacteria? They determine whether a bacterium can decarboxylate lysine to cadaverine, which would raise the pH. It helps to differentiate members of the Enterobacteriaceae and other gram-negative rods based on their metabolic capabilities. Um, the procedure involves using a needle to stab the butt and streak the slant. The butt tests for the lysine decarboxylation under anaerobic conditions, and the slant may show lysine deamination under aerobic conditions. The dual inoculation allows the direction of both pathways. The color changes that we should see are a positive result, resulting purple, indicating an alkaline pH because of the cadaverine production. Negative results are yellow, indicating that acidic pH from glucose fermentation without the lysine decarboxylation. Um, all of our results seem to be a bit skewed because none of them really change color. The only one that maybe there's a slight tone shift is for unknown 292, which appears to be a sort of pinkish towards the bottom. So I don't know what could have gone wrong for our results. Maybe we didn't streak enough bacteria. Maybe our bacteria was contaminated, or who knows. But what we should have seen for the E. coli was a purple butt, which we do, because it's a positive for lysine decarboxylase, and it'll decarboxylize late lysine, and it confirms the coliform identity. For morgani, it's a negative, and it should have been yellow. P. mirabilis should have been also yellow, meaning it's a negative because both of those do not decarboxylate lysine. For the S typhimurium, it should have been purple also, which it is, and it has a strong lysine dark decarboxylation. For the ornithine decarboxylase, these are results. The purpose of this in gram-negative bacteria is to detect the ability to decarboxylate ornithine to putrescine, which would raise the pH, and it helps differentiate enteric bacteria based on their amino acid metabolism. The procedure involves just looping the broths for an anaerobic condition. Um, Purple indicates a alkaline pH due to putrescine production, and negative results are yellow, indicating acidic pH from glucose fermentation without ornithine decarboxylation. Organisms that test positive uh, or negative for ornithine decarboxylase are E. coli, and Morgani and P. mirabilis, but they're variable. And negative is C. frundi. From our results, you can see that our E. coli is purple, meaning it is a positive because of the decarboxylation of the ornithine, which would confirm the coliform identity. Our C. frundi is a yellow broth, meaning it is a negative because it does not decarboxylate ornithine and it helps rule out other enterics. For our unknown 292, we see it is a purple color, meaning it is a positive ornithine decarboxylase test.
In conclusion, our findings show that the positive decarboxylase activity was indicated that there was an enzyme presence and amino acid utilization, and then our negative results suggested the absence of the specific enzyme or an inability to metabolize the amino acid. And so these tests are very significant because they help differentiate gram-negative bacteria based on their metabolic capabilities, and it also improves the diagnostic identification accuracy. Thank you so much for listening to our lab report. This is once again Group 2, Diego Castillo and Rita Cantu.